Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's start with a thought experiment. What if, one of your body's most incredible self-healing powers just stopped, like completely shut down, even after the thing causing the damage was gone? I'm talking about the liver here. It's truly this amazing organ famous for its ability to regenerate, right, to bounce back, even from pretty serious injury. I mean, it can regrow, even if a big chunk is removed. It's kind of a biological superpower. But, and this is the tough part, for millions of people who are dealing with alcohol-associated liver disease, or ALD, that superpower, it just fails them. And it's serious. ALD is the top cause of liver-related deaths worldwide. Often the only hope left is a transplant. Can you imagine how devastating that must feel, knowing your body isn't healing itself even after you've stopped drinking? So today we're diving deep into some really groundbreaking research. It's a new study, just published, that starts to finally explain why this happens, why the healing stops. And more than just explaining it, this work offers some real tangible hope for new ways to treat this.
It really is one of those big medical mysteries. And you're right, the liver's regenerative power is pretty much unique. We take away a big piece, and in a healthy person, it often just grows right back. But with alcohol-associated liver disease, that whole natural repair system just breaks down, it fails, and that failure leads to ongoing damage and eventually liver failure. We're talking, I think the number is around 3 million deaths every single year globally. Just think about that for a second if you're listening. The frustration, the despair, really, of having this vital organ that simply refuses to mend itself, even when the original cause, the alcohol, is gone. That failure to heal is exactly what makes ALD so severe.
So what is happening to those liver cells then? Why can't they fix things? This new research from teams at the University of Illinois, Duke, and the Chan Zuckerberg Biohub in Chicago, well, they found something fascinating. It seems the liver cells get trapped, they try to regenerate, they start the process kind of reverting back to a more basic, almost fetal-like state, which is actually a necessary first step for rebuilding. But then they just get stuck there, frozen in the middle. They can't complete the journey back to being mature working liver cells.
Exactly. The researchers who led the study, Avinash Kalsotra and Anna Maidil, they call it a quasi-progenitor state. Think of it like hitting pause halfway through a really crucial process. These cells aren't fully functional adult cells anymore, so they can't do the liver's vital jobs like detoxification. But they're also not properly dividing and multiplying to replace the damaged tissue either. They're just stuck in this unproductive limbo.
Wow.
And this creates this terrible feedback loop, a vicious cycle that just keeps pushing the liver further towards damage and failure. It's not just a temporary hiccup. It's a fundamental breakdown in the whole healing mechanism.
Okay, that's a huge piece of the puzzle.
Yeah.
But here's where it gets really interesting, right? What is physically stopping them? What's the molecular switch that's jammed? How did the researchers figure that part out? Because you can't just, look at a cell and see it's stuck.
Right. You need some pretty powerful tools. They used some very sophisticated techniques. They took liver samples from healthy individuals and compared them directly with samples from patients with severe ALD, thanks to collaboration with Johns Hopkins University Hospital. And they used methods like deep RNA sequencing and what's called multi-omic profiling. Think of it as creating incredibly detailed maps of thousands of individual cells simultaneously.
Okay.
This let them see exactly what instructions the cells were reading and how they were acting, comparing the healthy liver cells to the diseased ones. And what popped out was a major issue with something called RNA splicing.
RNA splicing. Okay, can you break that down a bit?
Sure. So think of your DNA as the master library of blueprints for your body. RNA is like a photocopy of a specific blueprint needed to build something specific, say a protein. Now, RNA splicing is like the editing step. Before you use that photocopied blueprint, you need to cut out irrelevant notes or instructions and maybe stitch together the crucial parts correctly to build the final product, the functional protein.
Got it. Like editing a document.
Exactly. But in ALD, the study found that this editing process, the splicing, was going wrong, broadly wrong, across thousands of different genes, thousands of different blueprints. They described it as being misspliced broadly. So thousands of proteins are potentially being built with faulty instructions.
Okay, so the editing process itself is broken. Is there a specific reason why? Like, is the editor missing?
That's precisely what they found. The main culprit seems to be a shortage of a specific protein, one called ESRP2. ESRP2 acts like that master editor. Its job is to grab onto the RNA copy and make sure the splicing happens correctly, cut here, stitch there. Without enough ESRP2, those edits don't happen properly, and the final proteins get built based on flawed instructions. And here's maybe the most crucial insight, a real aha moment from the study. It wasn't just that proteins were being made incorrectly, it was about where they ended up in the cell.
What do you mean where they ended up?
Well, because of the faulty splicing instructions caused by the lack of ESRP2, key proteins needed for regeneration were getting stuck out in the main part of the cell, the cytoplasm.
Okay.
When they actually needed to be inside the cell's control center, the nucleus, to do their job.
So they're present, but they're in the wrong location to function.
Exactly. It's like having the right tool, but it's locked in a shed across town when you need it in the workshop. It completely changes how we think about the problem. It's not just missing parts, it's parts in the wrong place.
That's fascinating. So it's a location issue triggered by this splicing problem, which is triggered by the missing ESRP2, a whole chain reaction.
Precisely.
Yeah.
And this ESRP2 deficiency has knock-on effects. It messes up other crucial proteins downstream.
Can you give an example?
Yeah, absolutely. Take a protein called SLK. Normally, it helps regulate cell growth and structure, and it's found in the nucleus. But when ESRP2 is low, the RNA instructions for SLK get misspliced, and the result, the SLK protein ends up out in the cytoplasm instead of the nucleus.
Okay, another protein in the wrong place. What does that do?
Well, this misplaced SLK in the cytoplasm then goes on to overactivate another protein called polo-like kinase.
And that sounds potentially bad.
It is. While you might think activating cell division is good for regeneration, in this context, it's uncontrolled and faulty. This overactivation can lead to something called mitotic catastrophe.
Mitotic catastrophe. That sounds dramatic.
It means the cells divide incorrectly, creating more damaged or dysfunctional cells, or they just stop dividing altogether, entering a state called senescence. So instead of healing, you get faulty construction, or construction grinding to a halt.
Wow. So the attempt to divide just makes things worse. Is there another example of this kind of domino effect?
Yes, another really important one involves a protein called TGF-β. This one is critical for a communication system in cells called the WNT pathway.
WNT pathway. I've heard of that.
Yeah.
Isn't it important for development and stuff?
Exactly. It's vital for liver development, for normal liver regeneration, and also for maintaining the liver's different functional zones. Yeah, think of the liver having different neighborhoods, each specialized for certain tasks. TGF-β helps maintain that organization. But just like SLK, in ALD, TGF-β gets misspliced because of the lack of ESRP2. And guess what? It also ends up stuck in the cytoplasm, unable to properly participate in that WNT signaling pathway in the nucleus.
So the WNT pathway gets weakened.
Severely weakened. And the consequence of that is what the researchers called a general loss of zonal identity.
So the neighborhoods lose their specialization.
Pretty much. The cells kind of forget their specific roles and where they belong.
Yeah.
It's like a city where all the districts blend together and nobody knows their specific job anymore. This massively impairs the liver's overall function and its ability to coordinate healing. How can it work properly if the organization breaks down like that?
Okay, this is incredibly detailed. We've gone from stuck cells to bad RNA editing to missing editor protein ESRP2 to other proteins ending up in the wrong place and causing chaos. But what starts it all? What makes ESRP2 disappear in the first place?
The ultimate upstream trigger. The research points pretty clearly to inflammation.
Inflammation. Okay, how does that connect?
Well, when alcohol initially damages the liver tissue, the body's natural response is to send in immune cells and support cells to the site of injury. It's trying to clean up and repair.
Makes sense.
But in chronic situations like ALD, this immune response becomes excessive and prolonged. These immune cells, especially certain types called T lymphocytes, start releasing large amounts of inflammatory signals and growth factors.
And one of those signals is the key.
Yes, one particular factor seems crucial here. TGF-β, transforming growth factor beta.
TGF-β. And what does it do?
This is where that vicious cycle really locks in. High levels of TGF-β released by those immune cells directly suppress the production and activity of our key editor protein, ESRP2.
Oh, wow. So the inflammation itself stops the editing process needed for repair.
Exactly. The body's attempt to respond to the damage inadvertently shuts down the machinery needed for proper healing. TGF-β also pushes liver cells towards something called epithelial to mesenchymal transition, basically making them less like functional liver cells and more like scar tissue precursors.
So let me see if I've got this. Alcohol causes damage. Inflammation kicks in. Inflammation releases TGF-β. TGF-β suppresses ESRP2. ESRP2 deficiency causes RNA missplicing. Missplicing leads to proteins like SLK and TCF4 being in the wrong place. This prevents proper cell division and disrupts liver organization. Healing fails, leading to more damage, which causes more inflammation.
You've got it. It's a self-perpetuating cycle of damage and failed repair driven by that inflammatory response.
That sounds incredibly bleak,
Yeah.
but the point of figuring all this out is, well, hope, right? Understanding the why must open up new ideas for how to fix it.
Absolutely. That's the real promise here. Understanding this detailed mechanism gives us completely new targets for potential therapy.
Like what? What are the possibilities?
Well, several things. First, those specific misspliced RNA molecules themselves, they could potentially be used as diagnostic markers, maybe allowing us to detect ALD much earlier.
Okay, better diagnosis. What else?
Second, we can now think about therapies to directly target that upstream trigger, the inflammation. Specifically, finding ways to block or reduce the effects of TGF-β signaling, since we know it suppresses ESRP2.
Right. Stop it from shutting down the editor.
Exactly. And third, perhaps the most direct approach, could we develop ways to actually fix the RNA splicing errors themselves, or maybe boost ESRP2 levels or activity, essentially intervening directly in that broken editing process?
Did they actually try any of this in the lab? Particularly the TGF-β idea.
They did, and the results were really encouraging, actually. In cell cultures, when they treated the liver cells with a drug that inhibits the receptor for TGF-β, it actually reversed the problem. ESRP2 levels went back up, and the splicing of those target RNAs, like for SLK and TCF4, went back to normal.
Wow. So blocking the inflammation signal allowed the editing process to restart correctly.
In those cell models, yes. It's a very promising proof of concept. It shows a clear, druggable target.
Now, obviously, cell cultures aren't whole livers, and mouse models often don't perfectly replicate human disease, right? There are limitations.
Of course, yes. The researchers are clear about that. Current mouse models for ALD tend to show milder disease than humans get, and cells in a dish can't capture the full complexity of the organ. But, as Dr. Kalsotra put it, these findings are a crucial launching pad. They provide the detailed map and the mechanistic understanding needed to design and test new therapies in future clinical studies. It's a massive step forward.
It really feels like it. We've gone from the general idea of the liver losing its superpower down into the weeds of RNA editing and proteins being in the wrong cellular zip code, all triggered by inflammation. And coming out the other side with potential new ways to intervene to actually help the liver heal itself again. That offers incredible hope beyond just transplantation.
It absolutely does. And it really highlights how crucial this kind of fundamental biological research is. Understanding these intricate, tiny details inside our cells is often the very first step toward finding real solutions for devastating diseases like ALD. Knowledge really is power here. Knowing why the healing fails gives us the power to start figuring out how to restart it.
So the final question maybe is, what will it take to really teach the liver how to heal itself again? It seems the future might involve not just removing the initial injury, like alcohol, but actively guiding those cells back from that stuck quasi-progenitor state.
Precisely. It might involve fine-tuning the inflammatory response or directly correcting those molecular editing errors. It really underscores how understanding the most fundamental processes of life can lead to breakthroughs for conditions that once seemed almost intractable.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.